Hey everyone, we are going to be doing a walkthrough through our first virtual lab that we are doing this year. So for this particular lab, we are going to be looking at the nervous system, and specifically the question for this lab is, how do leeches feel? So no, I'm not talking about emotions. I'm sure leeches might feel like happy or sad sometimes, but I'm talking about how they physically are able to pick up their touch sensation, right? So we are talking about sensory neurons. These are your nerve cells that actually pick up one of your five senses. So you have touch sensory neurons like in your fingertips and all over your skin. You're going to have taste sensory neurons on your tongue, smell sensory neurons in your nose, vision sensory neurons, they pick up light in your eyes, and you're going to have sound sensory neurons in your ears to pick up any sound coming in. I think I got all the five senses. So specifically today, we are looking at the touch senses inside of a leech. So when you're working through this lab, you should have both your lab notebook open and the website open at the same time. So on the cover here of our lab notebook, I have a picture of a leech. This is what a leech looks like. It is a disgusting little slimy slug looking thing that lives in ponds. And what they do is they will find you when you're in that pond and like attach themselves to you and suck your blood. They're kind of like vampire worms. Back in the day, fun fact, back in the day, doctors used to use leeches to suck the bad blood out of people, and they thought that would heal them. So this was a treatment for the bubonic plague, or like the Black Death plague. Doesn't actually work, but interesting concept. And then over here, I also have a diagram of a neuron, just as a reminder to what it looks like. So the directions tell you the same thing I'm about to tell you now, but you click here to open up this tab right here. It's also linked in Google Classroom for you. Read all the descriptions and instructions on the website and follow the website's instructions. Here we are. And then check back to your lab notebook to record your work. And I'm saying this right here, this slide deck, it is your lab notebook. So again, fun fact, if we go into presentation mode with this, and yes, I am proud of myself for doing this, it will interact with you just like a notebook will. So we have all of these different tabs here. If I want to jump to a specific part of my notebook, I can click it. So like materials, I click it, it brings me right to it, and I can go through my materials section. Now, let's say I'm done with materials. I want to check out the constant control, independent variable, and dependent variable. Again, right here, ready to go. Yes, I am proud of myself. You're welcome. All right, so we just read the directions. We have to start every experiment we do with a question, because remember, we're doing experiments to try to find answers about how the world works or how our bodies work. So the question we are starting off with is, how do leeches feel? And that's what it says right here. So I'm going to start reading what it says on the website, and we're going to look at it together. It says, why is there a leech in my lab? And also keep in mind, as you're going through this on your own, if I move my mouse over these underlined areas, it gives me a little description of whatever that is if I need it. So for a leech, it says the large medicinal leech, and that's its Latin name right there, is a useful model for studying the nervous system. Actually, you know what? I'm going to try to say that. Hirudo medicinalis. Yes, there it is. So the medical leech, or the large medicinal leech, is a useful model for studying the nervous system. Its nervous system is simpler and easier to understand than our brain, yet shares many mechanisms with it. So it works the same way, it's just smaller and more simple. Many important discoveries about the nervous system were first made in similarly simple animals like slugs, worms, insects, and squids. Very cool, interesting information. Anyway, in this lab, you're going to investigate the nervous system of the leech. You will use electrodes to record how individual neurons in the leech respond to different kinds of touch stimuli. So we're going to be poking the leech's sensory neurons. We're going to see it have an electrical response, and we're going to be looking at what that actually means. These neurons are large, making it relatively easy to record their electrical signals. By recording signals from different types of neurons, you can map how the leech's nervous system represents touch sensations through labeled lines. So what is a labeled line? Because we haven't talked about that in class. So it says it's the concept that each neuron carries a specific type of information. For example, one neuron may carry touch information from the left big toe. When this neuron is active, the brain knows that something is touching the left big toe. So a labeled line is basically just like an individual neuron's connection to the brain. All right, cool. Let us keep going. So it's telling us that in this lab, it's divided into two sections. First, we're going to have to dissect the leech. We're going to cut it open, see what it looks like, and then we get to probe and identify. So we're going to touch it on its different sensory neurons, and we're going to identify what type of sensory neuron we are probing. So to begin, press the leech dissection or probe and identify. We're going to do leech dissection. All right, so before we get too far into this, let's take a look over here. So what I'm showing you, and I'm going to zoom in on this so we can see it better. We have a couple of helpful diagrams. We have the anatomy of a neuron. So this is a neuron cell. We can see all of its labeled parts. We have our dendrites, our soma or cell body, the axon, which is wrapped in its insulated myelin sheath, and then we have our axon terminal or terminal buttons. These images right here, this one, yeah, I'll leave it on this. This one is showing us the type of reaction we're going to see when we poke the neurons of the leech. So the electrical impulse signal that we are recording is going to look kind of like this. We're just going to see some like lines on a screen. It's going to create a certain pattern. So similar to how there's like 2 humps here and there's 3 humps elsewhere, you're just going to see a slightly different pattern. And this is showing you sort of like how it works. So if we're looking at our taste sensation, here are our taste neurons, and it sends a signal. The electrical signal looks like this right here. It sends it to our central nervous system for our taste. This picture over here, let me move my face. This picture over here is showing us what this looks like inside of our brains. Remember, our peripheral nervous system, our sensory neurons, are connected to our central nervous system and our brain. So the sensory neurons have to pick up the information, and then it sends that information to our brain in order to process that information. So in particular, our touch neurons go to this area of the brain right here, this blue section of the brain right here. So here's the front of the brain, here's the back of the brain, here's the bottom, here's the top. We are looking in this blue section right here. Now, if we cut that brain, take out that slice, this is that slice of brain we are looking at. So this is showing you where all of your touch sensory neurons connect to in your brain. So if we're looking at your fingers, if you have a touch sense in your fingers, it's going to this part of your brain to be processed. The other thing to note about this picture is I know that these images look kind of funny. So we see like the fingers here, we see like the forearm and the elbow, and a lot of them are out of proportion. So like we have a giant head with giant lips, we have a giant hand, tiny skinny little arm. You guys get the idea. But what that's showing you is the bigger the image, the more brain space is dedicated to that area. So the more sensitive that part of the body is. When I say sensitive, I mean there are more neurons in that area of the body. So we can actually, I guess it's a more specific type of sensation. So this is the idea that in our fingertips, there's a large portion of the brain dedicated to this area because our fingertips are very sensitive. You can see this when people are blind and they have to learn to read Braille. They touch those little dots and they're able to figure out the differences between those dots so they can read the different letters in the words. So that's what this picture is showing you right here. All right, let's move on to our hypothesis. Yes, go. All right, let us zoom out back to fit. Cool. So now that we kind of have an understanding of what's happening in this lab, we're probing the leech and we're seeing how it responds with its electrical signal. We have to create a hypothesis to say what we think will happen. Remember, your hypothesis does not have to be correct. It is okay if you guess incorrectly, but I'm asking you to take that guess. So it says, if the leech's sensory neuron reacts when probed, so if we see that electrical signal, like this, that's the reaction we're looking for. If the leech's sensory neuron reacts when probed, then what does that tell us? So maybe, oh, I just zoomed. Go back to fit. There we go. Maybe we want to write in, if the leech's sensory neuron reacts when probed, then that neuron shows a touch sensation. It's your choice what you write in here. This is just an example, so you can use my example, you can create your own, but that's what's going down right here. So I suggest you pause the video, you make your own hypothesis. Ready? Cool. Okay, let's undo that and put it back to the way it was. Cool. All right. Now that we have our hypothesis, we have to look at our background information. So I'm actually going to come back over here. Oh no, this is getting into the procedure section. I'm sorry, I lied. Let's look at the background information so that, again, we can remind ourselves of some of the information about the nervous system that we already know. So question number 1 says, describe the function of the nervous system, okay? If you don't remember the function, you can always come back to the slide deck that was given to you about the nervous system and about body systems. So if I scroll through this slide deck right here, and I can find the nervous system, I hope I can. Here it is. It actually tells us the function right here. So I'm going to copy and paste. I mean, these are my own words, so I am allowed to use them. Right? So describe the function of the nervous system. Maybe I want to make it a full sentence. So the function of the nervous system is to regulate body processes, which makes sense because this is how our body communicates with our brain and how our brain communicates with our body. So then it says, describe the structure of a neuron, and you may insert an image. So all I'm going to do, again, I'm doing this because this is my work, so I'm allowed to use my work. But I am going to copy and paste this diagram of a neuron into my background information section. I'm just going to drag it over to the side for now, just so it doesn't get in the way of the words. But here's an image that I can add of a neuron. So let's discuss how, or just as describe the structure of the neuron. So we just have to list the parts of the neuron. So a neuron includes dendrites, a soma or cell body, an axon with an insulated myelin sheath, which I always spell wrong, myelin sheath, and terminal buttons. So I just listed the different parts of the neuron because in part 3 over here, oh, I said buttons, not buttons. There we go. For number 3 over here, it says describe the function of a neuron. So again, if you forget what a neuron actually does, let us scroll through this slide deck and figure it out. Here it is, neurons. So it gives us the definition right here. It's a specialized cell that makes up the nervous system, and its job is to pass messages or signals between the brain and the rest of the body. So I'm going to copy my own words again. And I'm going to say the function of a neuron is to pass messages. So just edit the grammar. Pass messages or signals between the brain and the rest of the body. Awesome. Cool. What is a labeled line? Ooh, we have to go back and check. Let us go look over here for what a labeled line is, because it said it on this previous slide. Labeled line. It is the concept that each neuron carries a specific type of information. So a labeled line is the concept that a neuron, that's not how you spell neuron, neuron carries a specific type of information. Cool. And then why are we using a leech in this experiment? Again, it tells us, let me move my face out of the way. So why is there a leech in my lab? In this lab, you are going to investigate the nervous system of the leech. You will use electrodes to record how individual neurons in the leech respond to different types of stimuli. And then it says, these neurons are large, making it relatively easy to record the electrical signals. So copy. I'm going to say, full sentence. I'm going to turn the question around. So why are we using a leech in this experiment? We are using a leech in this experiment because, and paste, it pasted in white. So let me turn that into black so that we can see it. Black. Cool. And fix the word because. All right, we are using a leech in this experiment because these neurons are large, making it relatively easy. Oh, I missed some. To send electrical signals, or to whatever it says. To record their electrical signals. There we go. All right, so now we have our background information. We've looked at these questions. We should have a better understanding of what it is we're doing and why. What I'm going to tell you is for this materials section right here, I'm going to have you do this on your own time. You're just writing down absolutely everything that this experiment uses. So you definitely need to include a leech because we're going to be dissecting a leech. We're probably going to cut it open with some scissors. We're probably going to need some sort of like electrical probe machine to pick up the signal. You get the idea. All right, so for the procedure, I'm going to be going back and forth between the lab and my lab notebook to write down what it is we're supposed to be doing as we do it. All right, so let us move forward and begin leech dissection. Here we go. Are you guys excited? Let's make this big so we can see it. So it says, first the leech is anesthetized. Basically, we knock it out using 8% ethanol solution. So we're using an alcohol solution. Ethanol effectively anesthetizes worms, including leeches and other small animals that breathe through their skin. However, concentrations of ethanol that are too high may harm or kill the animals. So basically, this is like if you put like chloroform to a human's nose and mouth, it just knocks you out. Why we're knocking it out and not like killing it straight out, I don't fully understand, because we're cutting it open to dissect it, which means it's going to die anyway. But here's the picture of your leech, and this is where it's going to be going. So let's take our first step, and it says the leech is anesthetized in 8% ethanol solution. So I'm just going to, again, copy and paste from the website and paste it right here, and now it's in white. Let's make it black. Cool, now we can see it. All right, so let's go to the next slide. All right, step 2, the anesthetized leech is pinned through the dorsal side up on a dissection tray. So dorsal side is the upper side. So for us, this would be our back side. And then it says ventral, which it doesn't say on this side. Ventral would be like belly side up. Dorsal is back side up. And it says the pins are placed through the leech's anterior, which is the front side, and posterior, which is the rear side. What does it say? The pins are placed through the leech's anterior and posterior suckers to stretch its body out. Okay, so this is a little bit different than humans. Anterior for the leech is going to talk about the head. Posterior is going to talk about the bottom. Anyway, so step number 2, we got to pin the leech. So again, I'm going to just copy and paste from the website. You can too. Are we there? Yes, so let's turn this black again. Cool. All right, so again, this is what we're doing. We got our leech, we're pinning it, right? Now, it says the skin is cut open along the dorsal midline. This is our step for step number 3. So again, I'm going to copy and paste. Did that paste? Yes, it did. The skin is cut open along the dorsal midline. Since the leech's nervous system is on the ventral side, so that's going to be on the, which one? I always forget. I confuse them. Just says underside. Sorry. So that's going to be the belly side. Since the leech's nervous system is on the ventral side, care is taken not to damage deep structures. The blood you see is both its own as well as its last meal. So let's come here and look at the pictures, right? So here is our leech. We're going to cut it open, which we can see with our scissors. And we keep cutting it, cut, cut, cut. And then when we open it, this is what it's going to look like. So we see some blood in here, which they say is from the meal of the leech. Remember, leeches eat blood. And then we see some other like red and pink in here. That would be the blood that actually belongs to the leech itself. All right, number 4, it says using fine dissecting instruments, the skin is carefully pulled open along the cut. So here is our next step right here. So again, copy and paste. Cool. The left and right halves of the skin are pinned down on each side. So again, let's look at what this looks like. Here it is open, and now it's showing us going in to pin it open so we can really start to see what it looks like inside of the leech. So again, what we're doing right now, this is its back that we've cut through, and what we're looking at is its belly. So this down here is the belly, this is the back. All right, so step number 5, the leech is now pinned open with the inside of its body facing up. You can see many of the internal structures, including its digestive, excretory, and reproductive organs. However, you cannot see the nervous system yet. It is located ventrally underneath the other organs. Okay, so it's just showing us what it looks like again. Cool. Obviously, you can pause the video and take like a closer look if you're interested, but I just want to get through all the stuff. So step 6, the internal structures are carefully removed, exposing a ventrally located structure called the blood sinus. So a blood sinus is a major blood vessel in the leech. That's what you're seeing right here. It's just like a big giant blood vessel. The blood sinus encases the leech's ventral nerve cord, which is part of its nervous system. So a nerve cord is a bundle of axons that connects ganglions to each other and the brain. So axons, we're talking about a bunch of neurons together, and then a ganglion is, again, just like a bunch of neurons put together. All right, so what do? We remove the internal structures. And again, let's make this black. Cool. All right, so it's also showing us right here. We're opening it up so we can see that blood vessel right here, and then we're going to zoom in over here to expose the sinus. So now we can really see that blood vessel. As you can see, it sort of had like a film over it that we had to cut through. All right, so step number 7, the blood sinus has many swellings along its length. So it's showing us right here. Each swelling contains a segmental ganglion, or if you're saying multiple, it's ganglia, of the leech's nervous system. So again, a ganglion or a bunch of ganglia is just a mass made up of cell bodies of neurons. So these are all the somas put together of a bunch of neurons. So here is a ganglia, ganglia, ganglia. So we can see, oh, here's another one. So we can see 4 ganglion, or no, ganglia. See, science is hard. Words. All right, step 8. The blood sinus is cut open using fine scissors, then carefully pulled apart with forceps to expose a ganglion. Here is our step. Let us put this into our procedure. And make it black. Y'all already know it's going to be black. Okay, there we go. You can now see individual neurons on the ganglion's dorsal side. Unfortunately, the neurons that respond to touch stimuli are on the ganglion's ventral side, so they are still not visible. All right, so let's look at the pictures. We see a ganglion right here. We cut through that to expose it even more. And then here is the ganglion right here with our nerve cords coming off in either direction. And we have some other nerve cords over here as well. All right, to access the ganglion's ventral side, the leech is flipped over. A small hole is cut into the ventral body wall to reveal the ganglion. Whoop, here's our next step. There we go. Cool. All right, care is taken not to damage the nerve cord or the nerves that come out of the ganglion. So this is, again, just showing us what this looks like. Here is our ganglion, our mass of somas or cell bodies for the neurons. We have a nerve cord, which is a bunch of the axons, and then we have some other nerves going on over here. Step 10, one section of the leech is isolated by making 2 parallel cuts across the body. The cuts are spaced far enough apart so that the section contains at least 1 ganglion. So again, I'm just going to copy and paste this into my procedure. And let's just highlight this all and say the color is black. Cool. Okay. So this is showing you we're cutting out this section right here, and you can see the ganglia or ganglion right here that we're going to zoom in on. All right, so the isolated section is stretched out with forceps, then pinned with the outer skin facing up. Oh, we're going to need some more space. Let us make this smaller so that it can all fit. 6. I'll make it bigger again at the end, don't worry. All right, I'm going to come back to that. Care is taken to avoid damaging the skin or the nervous system. The preparation is now complete. The ganglion should be visible through the hole in the section. Okay, so we did it. We made it. So now we're on to the probe and identify section. So again, let's make this all black so we can see it. And now let's make this a little bit bigger so we can see it. Let's try 9. Still too small. 12. A little bit bigger. 18. Too big. 14. Sounds good. All right, so we have our procedure for the leech dissection. Keep in mind, we have to do a procedure for probe and identify as well. I'm going to let you do this on your own too, just like how I did the leech dissection portion. So you can copy and paste directly from the website. So let's load up the results section because this is going to be the next part. So probe and identify sensory neurons. This section simulates a real neurophysiological experiment. So neuro, we're talking about the nervous system. Physiological, we're talking about the physical and chemical processes of neurons, okay? You can use your root words to look at this more closely if you are curious. So this section simulates a real neurophysiological experiment that explores the leech's nervous system. You may want to review background on the neuron, resting potential, action potential, and voltage display before starting the simulation. The only thing you need to understand is a neuron, okay? Resting potential and action potential, don't worry about too much. Resting potential, I will show you when we get to the next part, and action potential, I will show you when we get to the next part. Voltage display, I will show you when we get to the next part. And again, this is just sort of like what it's looking like in the lab. All right, so this is actually the 4 steps you need for your procedure for probe and identify. So copy and paste these 4 steps into your procedure, and then you will have your procedure done for probe and identify. So what we're going to be doing is through the following steps, you will explore how the leech's nervous system responds to different kinds of touch stimuli. Okay, so basically what you are seeing, this is your action potential right here. This is a visualization of what the neuron looks like. So neurons can have different structures. This is what this particular one looks like. And then we will be matching it to this chart over here to figure out what type of sensory neuron we're looking at. Here we go. Okay, so you are going to press start over regardless of if you're doing this again or whatever. Okay, start over. All right, I'm going to turn the sound off because it's an obnoxious sound. But basically, here we have a bunch of different neurons inside of this ganglia of a leech, right? So we've zoomed in on the ganglia, and now we're showing a bunch of different neurons right here. I'm going to click this one first, just for funsies, just as an example. So I click it, and this is its resting potential. This graph that you see right here is just a flat line. This is the resting potential it was talking about. Again, you don't need to worry about it too much. What we're going to do is look at these 3 different tools that we are using to probe our leech. So in your materials section, make sure you are writing down a brush as a weak probe, a probe as a medium probe, and forceps as a strong probe. Now, what you're actually going to do is click on it. So I'm going to click the brush, whoop, and it probes the leech for me, and it showed a response. So again, that was the action potential. In this case, there was a flat line, no response, but that was the action potential. I'm going to do the same thing with the medium probe. Click on it, it probes it for me. Again, no action potential, no response. And then last but not least, the forceps. Again, no action potential, no response. So now I press continue, and I'm going to click dye injection, which injects the dye into this particular neuron so that we can see it when we switch on the UV light. So this particular neuron right here, you're seeing the soma, the cell body, which is what the ganglia is made up of. And we can see its axon and dendrites going out to the rest of the nerve cord. All right, so we press continue, and now we have all of our information. So here is the visualization. Here are our different action potentials, and we need to match this information with the chart over here. So there's 5 different types of neurons. There's an N, a T, a P, an R, and an X neuron. And this is showing you the information that you need. So our resting potential, our no stimuli, there was nothing, right? So we're looking for a flat line that immediately eliminates this R neuron. When we look at what this neuron looks like, we need to match it with something down here. So for me in particular, it looks similar to both an R neuron and an X neuron. Now, we already know that it can't be the R neuron because for the resting potential, there's no reaction going on, but it can be an X neuron. So let's double check to make sure. So for each action potential, for the brush, for the probe, and for the forceps, there was no reaction whatsoever. And again, we see flat lines throughout this whole thing. So not only does the image match, but also our probing matches. So I'm going to click neuron X to identify it. And it says, good, you have identified the X neuron correctly. Yay, we did it right. Press the probe a different neuron button to test another neuron, which is what we're going to do. So I press OK, I press probe a different neuron, and I'm going to choose another one to probe as another example. Let's do this one right here. So again, resting potential, there is no reaction going on. I'm going to probe it with my brush. Oh, this is going to be a bad example. It's going to be the same type of neuron. We're not seeing any action potentials. Oh, we got one action potential. How interesting. So that graph will be recorded. Don't worry about it. I'm going to press continue. Again, I'm going to inject my dye. I'm going to turn the UV switch on. And this is what our neuron looks like. So it looks different than our last neuron. All right, so again, we have to match it. So I'm looking at the image here, and to me, this looks like either an N neuron or a T neuron in particular, right? So it matches either of these setups right here. Now we just have to match our different action potentials. So here, brush, no stimulus, probe, no stimulus, forceps, oh, that matches this one exactly. So it has to be an N neuron because the image matches and our action potentials match over here. So I press N. Good, you have identified the N neuron correctly. Yay, we did it right. Press the probe a different neuron button to test another neuron. I press OK, I press probe a different neuron, and you're going to continue doing this for all the different neurons in this lab. Now, after you have that, notice how it tells you the type of neuron right here. So we have N, we have X. These will fill in as you go as well, so that when you do come back over to your lab notebook over here, you are going to drag and drop these letters over to the circles that they match with. So this one right here is an X neuron. So what I'm going to do is take my X and drag it over here. And you can resize these little things right here to fit in your circle if you want. You can, let me move my face again. You can always zoom in on what we're doing so that it's a little bit easier to see what's going on. So now I have my X, I can make it smaller and make it like fit in the circle if you really want to do stuff like that. But that's how you're filling in your results section. Let me do this. All right, we're back. And let's go back to fit. Good. Okay. So after you do your results, we have to talk about the conclusion section. When we come back over here, I guess I'm just going to probe another one real quick just to get through. So boom, 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 continue. Turn them both on, continue. Okay, we have our information. It looks to me similar to this one right here. That is a P. I'm going to click P. Yes, I got it right. Perfect. So once you do all of them, you're going to click onto the conclusion. Yes, I am sure. And it tells me some more information, but basically you're going to use the conclusion section of the website over here as well as the rest of the lab to answer these conclusion questions over here. The last part I'm going to ask you to look at for this lab is the control, constant, independent variable, and dependent variable for this experiment. So I included some questions for you to help you think about what these 4 variables are for this experiment. But again, it's going to be your job to go in and try to figure it out on your own. So I hope this was a helpful little demo and explanation of how we're going to be going about doing this lab. At the end of the day, you are going to be turning in this lab notebook to me through Google Classroom to show that you completed the lab. If you do have any questions, of course, let me know in Google Classroom, by email, Google Voice, let me know during class. But again, I hope you found this helpful. Let me know if you have any other questions. And then good luck.